In genetics, a nonsense mutation is a point mutation in a sequence of DNA that results in a premature stop codon, or a nonsense codon in the transcribed mRNA, and in a truncated, incomplete, and usually non-functional protein product. It differs from a missense mutation, which is a point mutation where a single nucleotide is changed to cause substitution of a different amino acid. Some genetic disorders, such as thalassemia and DMD, result from nonsense mutations. Simple example. DNA, 5 feet ATG ACT CACCGA GCGCGA GCTGA, 3 feet, 3 TACTGA GTG GCTC GCGCTTC GACT, 5 feet, mRNA, 5 feet AUGACU CACCGA GCGCGA GCUGA, 3 feet, protein, MET-THR HISAR GALAG SER stop. Suppose that a nonsense mutation was introduced at the fourth triplet in the DNA sequence causing the cytosine to be replaced with thymine, yielding TGA in the DNA sequence. Since TGA is transcribed then translated as UGA, the resulting transcript and protein product would be DNA, 5 feet ATG ACT CACTGA GCGCGA GCTGA, 3 feet, 3 TACTGA GTG ACT CGCGCTTC GACT, 5 feet, mRNA, 5 feet AUGACU CACUGA GCGCGUA GCUGA, 3 feet, protein, MET-THR HIS stop. The remaining codons of the mRNA are not translated into aminoproteins because the stop codon is prematurely reached during translation. This can yield a truncated abbreviated protein product, which quite often lacks the functionality of the normal, non-mutant protein. Nonsense-mediated mRNA decay, despite an expected tendency for premature termination codons to yield shortened polypeptide products, in fact the formation of truncated proteins does not occur often in vivo. Many organisms including humans and lower species, such as yeast employ a nonsense-mediated mRNA decay pathway, which degrades mRNAs containing nonsense mutations before they are translated into non-functional polypeptides. The three stop codons have been given names: UAG is amber, UGA is opal, and UAA is ochre. OE amber was named by discoverers Richard Epstein and Charles Steinberg after their friend Harris Bernstein, whose last name means OE amber in German. The other two stop codons were named OE ochre and OE opal in order to keep the OE ameza theme. Stop codons are also called OE termination or OE nonsense codons. They signal release of the nascent polypeptide from the ribosome because there is no cognate tRNA that has anticodons complementary to these stop signals, and so a release factor binds to the ribosome instead. Pathology associated with nonsense mutations. Nonsense mutations can cause a genetic disease by damaging a gene responsible for a specific protein, for example, dystrophin in Duchenne muscular dystrophy. The same disease may, however, be caused by other kinds of damage to the same gene. Examples of diseases in which nonsense mutations are known to be among the causes include cystic fibrosis, Duchenne muscular dystrophy, beta thalassemia, Hurler syndrome. An experimental drug known as PTC-124 may be useful in treating some cases of each of the above diseases. PTC-124 was scheduled to enter the final phase of clinical trials in 2007. See also, nonsense suppressor, Emily's Entourage, a cystic fibrosis non-profit researching nonsense mutations. External links and references, Edgar B. The Genome of Bacteriophage T4, an Archaeological Dig. Genetics 168, 575 €82. PMC 1,448,817. PMID 15,514,035. References for the image are found in Wikimedia Commons page at Commons File Notable Mutations SVG References. Henderson, Mark. Daily Pill to Beat Genetic Diseases. The Times. retrieved October 23, 2007. Mutations, nonsense mutation, Gatfield D., Unterhälzer L., Ciccarelli F. D., Bork P., ALll E. Nonsense-mediated mRNA decay in Drosophila, at the intersection of the yeast and mammalian pathways. The EMBO Journal 22, 3960 70 doi, 10.1093 CDG 371. PMC 169044. PMID 12881430. Welch ALPTC 124 targets genetic disorders caused by nonsense mutations. Nature 447, 87 91. Bibcode, 2007 Nature 447. 87 W doi, 10.1038/Nature 05756. PMID 17450125.